But I'd like to welcome and thank you for joining us on today's OICR Mondays discussion event. We're going to begin with a land acknowledgement. We acknowledge the land that we are meeting on is traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabeg, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis peoples. We also acknowledge that Toronto is covered by Treaty 13 with the Mississaugas of the Credit. As this is a virtual meeting, we appreciate that many others may be on different territory, so please take a moment to reflect on and honor the Indigenous communities of your territories. My name is Michelle Brazis, and I'm the Associate Director of Adaptive Oncology, and together with my colleagues Shola Dukin and Jay Remnoth from Clinical Translation and Therapeutic Innovations, respectively, we have launched OICR Mondays, a discussion series highlighting research ideas and challenges along the cancer care continuum. This discussion series aims to gather the cancer research community to discuss contemporary issues in the field of cancer research, identify opportunities for collaboration, and brainstorm on innovative cross-disciplinary approaches that will lead to improved outcomes for cancer patients. So the format for today's session will be 3 short presentations from our speakers, followed by 30 minutes of discussion. So I'd ask that you please save your questions for the discussion period where you may unmute and join the conversation. Now I would like to introduce today's moderator, Dr. Martin Yaffe, who will guide us through the event entitled Imaging and Machine Learning in Breast Cancer Detection and Management. Dr. Yaffe is co-director of the Imaging Research Program at OICR, where he leads the Imaging Validation Core. He is a medical physicist and imaging scientist at Sunnybrook Research Institute and professor of medical biophysics at the University of Toronto. His research focuses on the early detection, diagnosis, and characterization of cancer. Martin, I turn the floor to you and stop sharing.
Okay. Thank you very much, Michelle. I'm delighted to be here. Please forgive any coughing. That's just kind of the end of my wonderful COVID experience. I'll try and do my best to suppress it. So we have 3 incredible presentations today, and as they used to say in Monty Python, now for something completely different. The talks really are quite varied, and they really will introduce you to the spectrum of how imaging and AI can have impact in our treatment, our management of breast cancer. So I just wanted to to set the stage. I wanted to show you this wonderful slide. This is the very first CT scan. It was done in England, published in 1971. This was one of the first examples of digital medical imaging. And the interesting thing you can see here is the pixels are very obvious. And there were this whole matrix of this brain scan, which revolutionized medical imaging, was 80 by 80 pixels. And things have only exploded since then in terms of the amount of data, what we can do with it. But right here, you see some of the examples of why digital imaging is important. And because this is a reconstructed type of image, as you can kind of see in the example shown in the upper left. And it's these applications of computation to imaging, facilitated by the fact that the images are in digital form, that's brought a lot of the power to the world of medical imaging. So what are the opportunities? And I'm sure I've missed many of these, but just for a start, we have to acknowledge that medical imaging really depends on the human eye and brain. Radiology, for example, involves very, very skilled people interpreting images. And the eye and brain are remarkable instruments, and they've evolved and specialized to be able to do what they do very well. But there's other things that they have not really learned how to do those things well, and that's where additional help may allow us to expand the capability and potential of imaging beyond what the eye and brain can do on their own. So examples where computation will be helpful is any kind of quantitative problem. Humans are not all that good at kind of turning things into numbers. They're much better at qualitative assessment. So biomarker analysis would be a good example. Structures that don't have well-defined edges, computers can often be used to try to do segmentation, find things where edges don't give you clues to where things actually are, where disease might be located. Looking at a second and higher order information like texture. And of course, when you have huge amounts of data, perhaps on different types of images, multimodality imaging, and when you're trying to integrate the data across different platforms and put them together. So you're going to hear additional areas from the speakers today of how some of the computing techniques and use of AI and advanced imaging can be helpful in taking us further in terms of our management of cancer, particularly breast cancer. A couple of examples that I'll just point to briefly, and these are just from the web, so they're really crummy images, are the use of computer techniques, algorithms, neural networks, and more recently, convolutional neural network approaches to the detection and diagnosis of cancer. This was straight out of the New York Times a couple of years ago. A technique that was actually partially pioneered at Sunnybrook is just pulling new signals out of fairly conventional images, again made possible because of the digital platform on which these images are acquired. In this case, using two energies, dual energy imaging at high and low energies, in this case of the breast, where you're injecting a iodinated contrast medium and being able to image the phenomenon of tumor neoangiogenesis and see tumors that otherwise might not be visible using conventional earlier approaches. And then finally, and this kind of leads into what you're going to be hearing from David Andrews and perhaps a bit from Anne Martel, is in biology, imaging is not limited to in vivo applications. Application in biology to look at biomarkers of cancer, better characterize disease, to be able to make better therapeutic decisions and do what we're now referring to as precision medicine, adapting the treatment so it specifically addresses a given cancer in a given individual by looking at, in this case, multiple cancer biomarkers. In this case, it's protein expression through a multiplexed immunofluorescence. So let's move on to our speakers. And first presentation, sorry, I've got to, whoops, just get to my program here. First presentation will be given by Dr. Anne Martel, who is at Sunnybrook. She's a professor in medical biophysics at U of T. She's now got my old job, the Torrey Family Chair in Oncology at Sunnybrook, and she's as an affiliate at the Vector Institute. And she works on in vivo medical imaging and digital pathology analysis. And I think you're going to hear about applications of machine learning in both of those areas for image segmentation, for diagnosis, and for doing predictive and prognostic work. Anne, it's all yours.
Great. I was just looking for the unmute button there, but I think I found it. So thanks a lot for inviting me to give a talk here. I will try and keep it more general and slightly less nerdy than my usual talks. And I'll try and run to time as well, though it's fairly tricky to do that. So before I start talking about applications of AI in digital pathology, I thought I'd just quickly review what anatomical pathology does in breast cancer at the moment. So here you can see a long list of all the different things that pathologists are expected to report upon when they're looking at either a tissue biopsy specimen just after a screening mammogram or a tissue resection specimen after surgery. So you can see, as well as looking at things like, is the cancer invasive or non-invasive, lobular or ductal, does it belong to a particular subtype, for example? Pathologists will also try and grade the tumor based on the type of cancer cells that there are, or whether a tubule is still there or not. So I'm just trying to get my laser pointer. Here we go. So first we try and grade the cancer, and then more increasingly common is things like doing immunohistochemistry imaging, where you will look at the receptor status, the HER2 new status. Some places do Ki67 score and so on and so forth. So a large amount of information that the pathologists are expected to report on. But more recently, there's been the opportunity to start digitizing these images. So slide scanners have been around for about 10, 15 years, and the AFI lab pioneered an early sort of home-built one as well. And what you're looking at here is a modern digitized whole slide image. So what you see here at the top is a thumbnail, and this is the whole area of tissue that we're looking at. And this black box here is the area that you're looking at in the larger view. And you can see a scale bar here as we zoom in and out of the tissue. The thing to note is that these images are actually gigantic. So I've put here one single whole slide image is the same number of pixels as about 10,000 mammograms. So the sort of tiles that we typically use for AI, we've got over 220,000 tiles in this image. And you can see that when we zoom all the way in, we can see all the way down to the nuclear structure. And then when we zoom out, we can look at the context. So we can look at the sort of macro structures in the tissue. So there's many applications of this, but obviously it means that we can now start to look at whether we can do some quantitative analysis of images. So applications, the first thing is obviously quantification and scoring. So things that humans do very badly. So pathologists, well, A, I think they don't like counting cells, and B, we tend to be quite bad at doing that sort of thing. So things like cell counting, scoring, assessing how much immunohistochemistry staining is going on and so forth. That's one of the main applications and things that have already made their way into some clinical workflows. The other big sort of win that people are looking at the moment is triaging and workflow management. So can we make it easier and quicker for the pathologist to find what they're looking for in these huge whole slide images? So for example, if we had an algorithm that could automatically detect micrometastases in lymph nodes, or if we could automatically find areas of abnormality on a whole pile of colonoscopy slides, for example, it would speed up the pathologist's workflow greatly. And there's some interest in looking at context-based image retrieval and diagnostic support. But perhaps more interesting to this group is the applications that we're looking at now in terms of prognosis and prediction. So can we take a digital pathology slide and look at some of these features that Martin mentioned that typically humans don't necessarily look at? And can we just make predictions and prognostic assessments? So can we predict what therapy a particular cancer will respond to and so on? And then if we can train artificial intelligence models to do prognosis and prediction, then can we look at where that model is paying attention to and then use that to think, well, if the model is looking at this area here, what's the morphological feature that it's looking at? And can that then go loop back to the biologist and then drive some discovery, which would be quite an interesting way of proceeding. There's a bunch of methods and tools that are being developed to support these applications. So the first one is detection. This is showing here a mitotic cell detection algorithm, which is one of the earlier things to make progress in. Once you've detected cells and outlined them, you can then start to try and quantify how much immunohistochemistry stain there is, for example, or you can count cells or find out how large they are and so on. We can do segmentation. This might have applications for looking for clear tissue boundaries and so on, or for identifying areas to then go and perform microdissection on, for example. We can do classification, although I'm not sure that this is one of the major applications for this, but sometimes an AI algorithm might be able to help, for example, in breast cancer, differentiate between atypias and non-invasive DCIS, which can be a very difficult thing for a pathologist to look at and take some experience. And maybe we can train algorithms to do that. And then finally, I've put here multiple instance learning / weak labels. This is more of a specialized technique where we don't have any detailed labels on the images. We just know the label for the image. So here is a lymph node, and this one doesn't contain metastases, and this one does. But this is the kind of technique we need to train for survival models, where we might know that, say, this patient had a recurrence after 5 years. This one didn't, for example. So these are all different kinds of techniques that need to be developed. But there are a lot of challenges in applying artificial intelligence to whole slide imaging. So as you saw from the first movie shot, that we've got a wide variety of scales, so all the way from low magnification, medium to high. And some problems you need the high magnification to see the nuclei detail. Sometimes you need the low magnification to look at the structures. So is the duct here, for example. But many times we want to actually look across the whole range of scale space, and that gives it particular challenges in designing the algorithms. Another thing that people don't often think about when they're thinking about digital pathology is the fact that we haven't got much diversity in a single slide. So I mentioned at the beginning that one slide has a huge amount of information in it, but much of that information is duplicated. So for example, this slide here may have hundreds of thousands of tiles in it, but most of them are just this boring fat area. And even areas of cancer in one patient might look very, very similar. But we have a huge amount of variability between patients. So all of these are patches taken from breast cancers, but they're all slightly different types of breast cancer. And as you can see, they all look very, very different. And our algorithms have to be trained to actually recognize all of these different patterns, which can be very difficult to arrange. And then things like annotations. If you want to get ground truth to train an AI model, it's very expensive. We have to get pathologists to annotate the images. We can't just ask members of the public. We can't use Mechanical Turk or anything. And even if we get experts to do it, we may get some unreliable results. So this is an example from a ductal carcinoma in situ project we have. We asked 2 pathologists to annotate the images, and we asked them to make cyan the certain that the duct is here. And then yellow was the, well, not quite so certain. And one of these was an experienced pathologist and one less experienced. And you can see that even with those same instructions, we have 2 very different sets of annotations here. Fortunately, there's been a big increase in the number of public, well-annotated data sets that have been made available. Chameleon-16 was one of the first. This looks at lymph node metastases detection. And this actually drove a lot of the interest in some of the big companies like Google, because they suddenly had access to very high-quality annotated data. MyDog is a mitotic cell counting challenge, and here the challenge is actually to make an algorithm generalizable so that if you train it on 1 or 2 scanners, it'll be able to generalize through other unseen scanners with slightly different intensity levels and so on. This is a more recent challenge here. This actually looks more at the prediction type of problem, looking at tumor infiltrating lymphocytes and scoring TILs. Vishwesh, one of my students, took part in this challenge. I was hoping to talk about that, but unfortunately I've not got time to include the slides from that. But this is looking at prediction prognosis. So I'm going to move on to some of the work actually done in my lab. So when I first started in this, I'm working quite closely with Sharon Norfolk-Moses, a breast pathologist at Sunnybrook. And she's very interested in the application of AI to breast pathology. So I asked her, well, what's a problem that you would want to solve? What would be a good clinically relevant problem that needs to be tackled with AI? And she immediately said residual tumor burden. So typically when a patient's had neoadjuvant chemotherapy, they have therapy, then they go to surgery, then the tissue is resected. And the traditional way of assessing response is a pathological complete response. So the pathologist looks at the tissue and says there's no cancer left or there is residual cancer. It's a very binary measure. But Simons came up with a more sort of quantitative tool, the residual tumor burden, which contains a component on looking at the extent of the residual tumor cells, the number and the density of the residual tumor cells, whether it's in situ or invasive, and whether there are lymph node metastases, and generated a scoring system that's very well validated in clinical trials, and is actually able to make a decent prognostic prediction. But the problem is it's very time consuming to calculate. So these are some numbers from Sharon here. And what she did was took about a dozen cases, I think this is just a few of them on this table here, and she looked at how long it took her to generate a residual cancer burden measurement. And you can see the time she spent in minutes and the number of slides. So each case would have many slides for the tissue block through the tumor, and the larger the tumor bed, the more tissue blocks there are, and also how many lymph nodes were resected. So you can see the maximum one here is 180 minutes with 46 slides. You can see this is incredibly time-consuming. This is not done as part of routine clinical practice in the community because it's just too long to calculate, but it could provide valuable treatment information to guide oncologists with how to treat patients. So great, we'll start doing this. So the first thing we tackled on that list of things to do was the tumor cellularity. So this is a measurement of, given the patch, how many tumor cells are there per unit area in the tissue, which is part of the calculation we need to do. We chose this because it was relatively easy to annotate. Well, not quick, but at least we could give some clear guidance on how to annotate it. So Shireen Salam, a research pathologist in my lab working under Sharon's supervision, was asked to take whole slide images, and we had about 100 of these whole slide images, and she would drop uniformly sized patches on the image, and then she was just asked to randomly sample representative samples of different levels of cellularity. So 0 is normal tissue here, no cancer cells. all the way up to 98% coverage of cancer cells, and the different colors reflect that. Fortunately, she also put a black boundary around to show us the extent where there were no tumor cells. So outside here, we know that's all normal tissue, and that will come important later. And we actually made this, we got permission from Sunnybrook to make this available to the public. We used it to organize one of these challenges, which is a great way of sharing data and getting other people interested in the problem. So we set up a challenge in 2018 in conjunction with the NCI and the AAAPM SPIE meeting. And we had 317 teams register. 41 of those teams submitted entries, so they actually managed to get some useful information out. And 21 of those teams were matching the pathologist performance. In other words, the interclass correlation coefficient between the pathologist and the algorithm was the same or better than the correlation coefficient between two separate pathologists. So they managed to do this task very well. And the nice thing about having a challenge is you can then interrogate your algorithm to see how well it works. We found out that all the teams did well on the easy patches, so no tumor here, high density of tumor here, but they do very poorly when different tumor types are not represented in the data. So this is actually normal tissue. It's just very densely packed epithelial cells. Here, the algorithms got confused with the lymphocytes. And this is mucinous carcinoma. This one's got a lot of cytoplasm. So these weren't really present in the training data. And then the next thing was quite interesting. We then wanted to look at the tumor bed extent. So naively, you'd think, well, you just run your cellularity calculation, do some post-processing, and draw a hull around the areas of tumor. So that should work very nicely. But the trouble is, it's very, very sensitive. So this is the problem when you go to whole slide images in clinical practice. So any small error will cause a huge difference in your quantification. So small false positive detection here will cause real problems when you're trying to actually assess the tumor bed extent, which is actually these two blue lines here. This is the metric that's actually calculated. So Ozan Siga was a student in my lab. He looked at this problem and realized that the problem was that when you're doing patch-based, you're very sparsely sampling the tissue with the patch-based method. So we're training our cellularity algorithms just using a few patches here, but we're not capturing the huge variety of tissue that's outside of those patches. So we're not capturing things like pen markings, blood, these areas of tissue here, all the fat and so on. So what he did instead was he sampled much more negative patches from the sample. And in order to stop just sampling the same thing again and again, came up with a novel clustering technique to do better sampling on that. So I'm going to have to rush through these because I'm running out of time already. But the take-home message from this work is that algorithms that are trained on patches, so the sort of classical old method of doing challenges where somebody curates a set of patches, works fine when you're testing and training on patches, but does very badly when you're actually deploying on whole slide images. So this line here where it says no negative sampling is the patch-based method deployed on a whole slide image. And the key thing here is on the confusion matrix, everything is a false positive. So we don't actually have any true negatives. Whereas if you use this k-mean sampling and did all the negative sampling, we actually get up to much better performance. So very important to realize that you can't rely on patch-based methods to do real clinical problems. Here. So moving on from that, we have a real problem in all of digital pathology. We have very small data sets. So one of the key ways of trying to overcome small data sets is to do something called transfer learning. And the idea here is you take a very large data set, and typically people use things like natural images, ImageNet, and so on, and you train a neural network of your design on this very, very large image data set. So you train an algorithm to recognize the difference between cars and cats and dogs and so on. And you assume that you're learning something important about the representation of the image by doing this. And then what you do is you transfer the weights into a new model, and then you fine tune it using your smaller labeled data set from the domain that you're interested in. But of course, trains and cats and dogs don't look anything like histopathology images. So we've done a lot of work on self-supervision, and this continues to be a main focus of my lab. I haven't got time to go into these methods, but some self-supervision methods, you generate synthetic labels from the data. So here we're looking at whether or not the resolution, what order the resolution patch is in, which takes advantage of the multi-scale nature of the data. Some methods just use self-supervision, so they just try and generate useful representations from the raw data itself. This was one project done by 2 students in my lab. They pre-trained a model using a huge number of data sets from the web. We released this model actually publicly. So this was a pre-trained Resonate 18 model, and at least 3 different papers in the literature I've seen other groups download this data and show that when you actually use this model, so this is an example of some downstream tasks here from data that wasn't in the model, you not only increase the performance, so the blue line shows you the accuracy with using our pre-trained model as a starting point, but you also decrease the amount of data that you need for training. And the other cool thing is once you get a good representation, you can start to do some unsupervised things like clustering. So here you've got some unsupervised clustering, and you can start to look at patterns in the data just by looking at what the appearance looks like and finding similar things. So just as a final slide, because I can see I'm over time, we're also looking at how to deploy these things. So this is an example of a dashboard that's been put together by a software developer in my lab, Lucas. And what he's doing is trying to make it so that the pathologist can push a button and it can go through a whole pile of images. In this case, these are hematopathology, immunohistochemistry staining for P53 mutations. And then we can show heat maps over the whole slide image. We can generate quantitative values looking at the whole slide image, or we can try and reproduce what a pathologist would do by trying to do some hotspot analysis and so on. And we're experimenting with different ways of presenting results to pathologists and so on. So this is work in progress. So finally, just to summarize some of the challenges. So when you're training AI algorithms in any area, actually, selection of varied training cases is very important. We can use lots of tricks like unsupervised pre-training, semi-supervised approaches, for example, to try and reduce our dependence on annotation. And things that we really need to start looking at are things like how do we do domain adaptation, deal with different machines, different centers, how do we interpret the results of these deep learning models, very important for pathologist confidence, and how do we detect out of sample cases? So how do we know if the model that we've trained isn't going to work on the particular patient population or the particular sort of equipment that's been used? So if we change X-ray equipment or slide scanners, can we still work? So just to wrap up, these are all the people I work with and who've done some of this work. So thank you very much.
Thank you very much, Anne. A real challenge with a short time and lots and lots of things to say. But it was great. We'll have time for questions and discussion later. Let's just move on to the second presentation, which will be given by David Andrews. And David is a senior scientist and he's director of biological sciences at Sunnybrook. He's also professor of biochemistry and medical biophysics at U of T and Canada Research Chair in Membrane Biogenesis. And he's going to talk today about the use of organoids for screening for new therapies for cancer, and I assume today breast cancer. David?
Thanks, Martin. And thanks everybody for coming. Thanks for the invitation and thanks for everybody to come. At the beginning, people had their cameras on and it was quite nice to see a bunch of people that I haven't actually seen in a long time. So
yeah, let's get those cameras on, folks.
Yeah, well, it's okay for now. Anyway, it was fun to see people that I haven't seen in a long time. So I am going to talk to you about organoid models, and not just for predicting patient responses, but the usual way that I introduce this topic is shown here, and it's the way that most people think about it, is that what we're trying to do is to take a sample from a patient and then use some kind of screening. In this case, I'm showing drugs to perturb that sample from the patient. And then we use automated high content imaging to record data from it. And the idea is to come up with the best drug or gene therapy for that individual patient. So a precision medicine kind of application. But in fact, precision medicine is just one part of this, and certainly a part that we're working on a lot in my lab. But we also use this to screen new drugs in an effort to not just to reduce animal testing, but to replace it with something that would work better. I'm also involved in a project with Novartis, in which they're actually now wondering about whether or not they should give up on mice altogether, because we've cured cancer so many times in mice and that it not translate. And we also use it for genetic screens to identify potential new drug targets. So we use it for all of these applications. Now, in organoids, we're moving away from 2D cultures, 2D cell cultures, to generating something that's growing in 3D. And people may remember a long time ago that this is very popular as spheroids. The difference between a spheroid and an organoid is that in organoids, you're using something called conditional reprogramming to put the cells back into cycle and allow them to reconstruct the kind of tissue that they come from. So the idea is to model the tissue rather than just have a bunch of disorganized cells stuck together. And we do mostly live imaging, which allows us to image the cells at multiple different time points. It does require special dyes. And then to do that kind of imaging that Anne was talking about earlier, the problems, the challenges are very similar. So some of this will already sound familiar thanks to her talk coming before mine. So the first question is, can we do this in high throughput? Because if we really want to get something for patients, we need to be able to do it fast, and we need to be able to do it in high throughput if we're going to test more than half a dozen drugs. And the answer is yes, it takes automation. But what I'm showing you here is 10%, just one corner of a 1536 well plate. And in this plate, each one of these little wells is just over 1 millimeter square. And in those, you can see that we've got organoids growing, and we're getting pretty good at getting them to grow close to the center of each of those wells. And in each of these cases, we put in 1,000 or so cells, and you can see we get several organoids that are coming out. So that this would be one organoid here. I'm going to point these out because I'm going to be changing scale as we go along. So this is now getting bigger. So here's your one organoid again. And now using live cell dyes, we can monitor the growth of these things over time. In this particular case, we're growing them in little cups made of hydrogels. Those hydrogels can be modified so that they mimic the extracellular environment that's normal for the cells. And so you can get these things to grow into this kind of organization. And I would say probably the most common use at this point for something like this is to do drug testing, where you're just going to try now, after working all that hard to get them to grow, you're going to kill them. And so what I'm showing here is an individual organoid. This is just one. And at low drug concentrations, you can see that it's very unhappy. And at high concentrations, it's all gone. I need to introduce to you here the colors again. So the yellow color is metabolically active cells. It's measuring the transmembrane potential of the mitochondria. The green is extracellular matrix and dead cells, and then the blue is nuclei. And nuclei are a particular challenge in this area because almost all of the segmentation algorithms that are available work on identifying nuclei and then identifying your cells from there. But we want to get a lot more information from these organoids than just are they alive or dead, or have we obliterated the cells. And this is now what these things look like at a bit higher magnification and growing over a longer period of time. So we've got day 7, day 22, and day 35. There's 2 different organoids here. This one is the same here and here, as is this one repeated here. You see that these things grow for quite a while, and they can get pretty big. The 3 colors again are the membrane potential of the mitochondria is yellow. A live cell dye that we developed in my lab that lets us see more senescent type cells is the red, and then the green is cells that are dying. And we can increase the magnification further as we were the same kind of problem that Anne was showing. This here, we're still only up to a 20x lens. When we get to a 40x lens here, now you can see that there's all kinds of subcellular detail. I can point out one cell here. If I look at just this one cell, you can see the individual mitochondria in it. And the other important thing to point out is that you can't see the nucleus. So this blue stain is the nuclear stain that people normally use for segmentation. and it doesn't penetrate very well inside the organoids. The other colors that you're seeing here are the dyes that we developed, which are not toxic, so they can be in throughout the whole growth of the organoid. So everything is stained well with the other dyes, but nuclear segmentation and then cell segmentation is a real problem. However, the other thing is that now you're also seeing that this is, just a little corner of one of these organoids. There's really an awful lot of data here. So how can we get drug responses from this? We don't just want to see whether or not we've killed the cell, but how they're actually responding. So this is another set of organoids. I'm sorry, the colors have all changed, and that's because the postdoc working on this is red-green colorblind, so the color scales have changed here. So the nucleus is still blue, and X and 5 is now purple. The mitochondrial membrane potential is orange, and what was red before is now gray. And so here's a nice organoid. It's growing. It's being treated with a control drug that doesn't kill cells. Like most tumors, there's lots of cells that are very unhappy, and these are dead over here. These are dying. You can tell by the nuclear condensation here. They are going to die. The purple is the ones that have already died, and here's the ones that are energetically alive. And this is very typical of a tumor also, that where you have lots of dead cells or dying cells. And we want to take this and then add an experimental drug to it. And just to show you how complicated this imaging problem is, this is now at 1 micromolar drug for 72 hours. You can see the nuclei are condensing over here. There's lots of these pinpoints that are turning white. There's now lots of an X and 5 staining, so those are dying. We're losing metabolic activity. The cells are senescing or starting to die. At 5 micromolar, the problem is much worse. And at 20 micromolar, pretty much everything is dead. And so the question then becomes, how are we going to get information about each of these cells? Because there's different numbers of cells in the organoids at different times. How are we going to parse this out into something that's useful? And it turned out that, as part of the reason it's appropriate for this talk, is that it was a deep learning neural network, which was what we used. And that was to identify the individual cells, but without using the nuclei as a fiduciary mark as to where a cell was. And this particular pre-trained neural network is called CellPose, and it allows us to take the data that you saw before and turn it into something that looks like it came from flow cytometry. So now this is density, because there's too many points to have individual cells shown. But the upper left-hand corner, those are the cells that are healthy and alive. As they move down, they're losing metabolic activity. And as they move right, they're getting an X and 5 staining, so they're becoming dead. And now we can start to see how the different drugs work. So the control drug isn't really changing the pattern as you go down over time. Thapsigargan, a drug that interferes with the ER calcium pump, moves things pretty much directly from the live to losing metabolic activity and becoming dead. Whereas storosporin, a kinase inhibitor, moves them more towards being dead without losing so much metabolic activity. And our experimental drug that we're testing for a local biotech company, you can see here, is just annihilating the cells in the organoid. So we get more information. We went from 1 parameter, are you there or are you not, to 2 parameters where we have metabolic activity and apoptosis as opposed to just gone. So we're getting a bit more information. But really, if we start to look at this in more detail, we want to get a lot more information. So what I'm showing you now is a pre-malignant organoid. So this is not yet cancer. And what we're going to try and do is to take this and do a genetic screen where we put genes in that cause cancer and ask what's the effect of these genes on the organoids. So full transformation there's a lot more information here and a lot of different cell types and a lot of things that we need to decipher. And as I mentioned before, segmentation is a huge issue. So our approach to this problem has been to create software that doesn't use cell segmentation. It segments the object and then uses a more of a shallow learning approach, because we need something that we can calculate relatively easily to analyze these images. So here's the pole problem laid out. So each one of these columns is a different gene that's been introduced into an organoid, and that's a single organoid. So this is the top, the middle, and the bottom. But in reality, there are 80 confocal sections between the top and the bottom. So there's a lot of data here. And then we have the different oncogenes across the top. Parental and empty vector are controls. And so the question is, can we tell the difference in the different oncogenes? And can we show that the automated analysis of these images is actually giving us something useful? So how do we analyze them? The first big problem from a machine learning point of view is that these are real cells and they spontaneously transform. And in the negative controls where we don't have transformation, we actually do in about 50% of the organoids. So dealing with that is also an issue when you're trying to do the machine learning. So this is the approach laid out. We start with our multi-channel stack. We identify pixel categories from this stack. So we reduce all of the different colors and levels of intensity to 50 or so different pixel categories and redo the images in those pixel categories. We then combine those pixel categories, those pixels, into larger three-dimensional structures, little blocks, kind of like the little cutouts that Anne was showing before, but these are three-dimensional. And then we do the same thing where we assign them categories. And then we take those supervoxels and combine them a second time to make megavoxels and again get categories. And in the end, we have a categorization of the original image by the megavoxel categories. So this is essentially now a multi-parametric description of the original image stack that we had over here. And you can use this multi-parametric description, it's only 45 parameters, to cluster or classify the different little volumes that you've extracted from the much larger 3D image. So how does this work and how do we know it works? What I'm showing you here is the results from the organoids that I showed you before. And these are the clusters that you come up with. We have a way of automatically assigning the number of clusters that we know over clusters the data. So we expect to have too many clusters, but hopefully not too many. And then here are the different oncogenes on this side. So the first thing that you notice is that the empty vector and the parental are quite similar in their distribution across the different clusters. And we can also use manual annotation to go back and find out that these two here, clusters 1 and 2, those are the normal ones, whereas 6 and 7 are the spontaneously transformed ones, which we could guess from the rest of the data, but we did go back and check. BCL2 and BCLXL are both oncogenes that prevent cells from dying, and they have similar patterns across the clusters. MYC and MYC T58A, T58A is a more transforming version of this MYC, but this MYC is expressed at high levels, so they're about equivalent biologically. And again, you get the same distribution of clusters for those two oncogenes. Snail is a different one, and it's different. P53, we have 2 mutants, one that's stronger than the other, and PI3 kinase is the one that I showed you to begin with that gives you that dramatic transformation, and it accounts for cluster 7, the ones that are highly transformed. So we get an answer from this analysis that fits the biology, the known biology of the different oncogenes that we put in. We wrote this originally in MATLAB, and now we've been working with Anne's lab, and this is completely rewritten in Python. It's undergoing final testing, and we're hoping to release this to the general public very shortly. So the beautiful thing about this is this was 1,300 organoids, 80 slices each, and could be analyzed in 1.5 hours on a laptop computer. So this is a really great way to explore your data in a very fast way before maybe doing a more rigorous or time-consuming analysis afterwards. Okay, so what's left is just for me to thank the people who've been working on this. As you can see, we're developing things and we're getting there. We're not completely ready for prime time, but we're moving that direction. I'm working with Lillian Jean, Helen Mackay, and Katarzyna Jerzak. We've now started a clinical trial, which is in triple-negative breast cancer and ovarian cancer. Those patients will be taking samples from prior to their treatment. We'll get a second sample after they get the first drug, which is a known drug for treatment. And we'll be taking those samples and predicting the outcome from the second drug, which is the experimental drug, navitoclax. And hopefully we'll be able to find out whether or not we can really have prognostic activity and be able to determine which patients will benefit from the treatment in real time, because it takes us about 2 weeks. I acknowledge the funding sources and the rest of the contributors that are working on this, and thank you for listening.
Great, thanks very much, David. As I said, we're going to be really hearing very different pieces of the story from the three speakers today. So to round this out, we'll move on to the final speaker, James Mainprize. And James is a research associate in my group in the physical sciences platform at Sunnybrook. And he's been very interested in digital mammography and breast tomosynthesis, right from the development of hardware and looking at risk-benefit modeling, but also in trying to optimize strategy for doing screening using a precision medicine approach. And today he's going to tell you about the work on that, which is actually supported as a pre-CATA project through OICR. James?
I also had to search for the unmute button. They hide. So thank you very much, Martin, and thank you everyone for joining us. I'm going to talk a little bit about the project that we've been working on, as Martin mentioned, in the pre-CATA program. And it's looking at a particular problem in screening mammography. And before I do that, I just wanted to just sort of indicate the team that we've managed to put together for this project. The pre-CATA, for those of you who may not be familiar, is the Preclinical Trial Acceleration Team Award that's provided through the clinical trial program at OICR. And what we're trying to do is build up essentially the evidence that we need to be able to move forward to a larger or a more formalized clinical trial. And the group you can see here, it's both patient partners as well as Canadian and international partners. So this project, we're looking at something called masking risk, and masking risk occurs in screening mammography. And what I've got shown here is examples of different mammograms shown here from women with different amounts of breast density, from the non-dense breast or fatty breast to a breast that's very, very dense, shown here. And basically, the density is the amount of fibroglandular tissue that's in the breast. And the unfortunate thing is that white density that you see in the image can obscure lesions. And so a lesion that might be easy to see, such as this little nodule here in a fatty breast, would be completely hidden in the dense breasts. And in these intermediate regions, depending where the lesion is and depending what kind of density it is, they may or may not be evident in that. So the result is that screening mammography, the sensitivity drops as you go up in increasing density, and somewhere around 85% in the fatty breast, drops down to somewhere around, say, 60% in the densest of breasts. So that such as the result is that, say, for example, in the Ontario breast screening program, if you take a look at the 2-year screening sensitivity, it's about 75%, meaning that we're missing about 25% of those cancers. And some of those are because they've been masked by that density. The unfortunate thing is that this sort of classification scheme, this A, B, C, and D for breast density, is assigned by radiologists. And unfortunately, that part of it it's qualitative, and the radiologists aren't very good. And mentioned that trying to quantify and classify is something that's very tricky for humans to do. So we've come up with essentially a masking risk score that is based on some of the radiomic features that we can extract from these images related to the breast density. And what it is the masking risk is identifying images that are essentially hard to read because of the tissue complexity that's in that density in the mammogram. And it really does outperform the radiologist's assessment of masking risk when they're using their 4-category breast density classification system. So what can we do with this? Well, the idea is if we can identify women who are at high masking risk, knowing that mammography isn't going to work very well for them, can we suggest a stratified or a personalized screening approach for those women? And that might look like something like this. This would be the workflow for a woman coming in for her semi-annual or biannual or annual screening visit, where they get a screening mammogram. They're assessed for masking risk from that mammogram. And women with high masking risk, meaning that they have a complicated mammogram, they could be reassigned to get additional or extra screening with a technology or an imaging modality that is less sensitive to the amount of breast density. So that might be something like ultrasound or MR. And the idea is that we'll find a few more cancers in these women, and that we find them earlier, and that we may be able to treat them more effectively because we found them earlier. And the result would be then an improvement in survival, not only for those individual women, but overall across the entire population. So this masking risk tool we've started to call Mammatus, and Mammatus was so named because in the past, and for a number of years, we've used a in-house software called Cumulus that is a quantitative breast density measure that measures the amount of breast density that's in a mammogram. And Mammatus is sort of its granddaughter or next generation version of that, of Cumulus. And it's so called because there are clouds that look like this shown here, where Mammatus is essentially this really interesting cellular pattern of pouches of cumulonimbus rain clouds. And so that seems to be a very appropriate kind of name for something that's looking at breast images. The masking risk score that Mammatus extracts is based on, like I said, the radiomic metrics that are related to both the amount and texture of the density, and it's something called detectability. And that's shown here in this little inset here, where detectability can be estimated or evaluated by inserting a artificial lesion into the image, and that's that little dot that's showing moving across the image, and calculating the signal to noise ratio of the lesion to that background. And the signal to noise ratio will depend on both on the amount of density that's in the background, as well as its textural features, so how it looks in that background. And that's just the reference where we've shown some of our report performance to date. So I should point out, though, I just bear in mind, so some of you may be familiar with things like computer-aided diagnosis. Mammatus is not a lesion detector. We're not finding hidden lesions or anything like that. What we're doing is assessing the background mammographic density and assigning women to either a high risk or a low risk for masking based on that texture, basically saying, is it a hard image to read or an easy image to read? Now, that could be combined with a CAD type program, and that may lead to improved performance above and beyond what we're talking about today. We have essentially two versions of Mammatus. One is, I'm going to call today the Mammatus ML, which is a machine learning model that relies on the more classic approach to machine learning. Sometimes it's referred to as the handcrafted machine learning. And this is where we, essentially pick a few parameters and generate models based on those parameters. And in this case, what we're doing is extracting radiomic features, both from the detectability map, which is the sort of the blue highlighted region over here, that essentially establishes lesion conspicuity in that background. Like I said, from the signal to noise ratio, and it's based on simulating lesions and inserting them into the image to calculate that value. We're also extracting radiomic features, and these could be the first order features like mean value, standard deviation of the pixel values, and so on, or higher order features such as the gray level co-occurrence matrix, which are much more complicated constructs, but then can lead to very interesting information that humans have difficulty both in perceiving and as well as quantifying. The idea here then is we extract these features and build up essentially a linear model. It's based on a logistic model where we're looking at the odds ratios of in a labeled data set where we have mammograms associated with cancers that were eventually missed versus mammograms that were associated with cancers were able to be detected, and essentially measuring or evaluating the odds of that happening with that given mammogram. And because this is a built-up model, we needed a way to select and pick the right and the most promising parameters. We used a penalized model building approach where we use something called the ACA-EK Information Criterion, or AIC. And what that does is it penalizes the addition of extra variables into your model. And the idea being there is you want to prevent overfitting. If we used all of the radiomic features, we could certainly get a very good model, but it's probably overfit and it's actually fitting to bad data or erroneous information. And so this helps to keep the model as simple as possible while also explaining as much of the model as possible. We also have Mammatus AI, which is a deep learning model, and Martel showed something like this, where you take a pre-trained network and essentially do some transfer learning to get it to work on, in this case, mammograms. And we also showed a very good performance with that. It's sort of neck and neck as to which one to choose. We tend to go with the ML version of this, and that's partly because in some of the simulations we find that the ML version of the Mammatus software seems to be slightly more efficient than the AI version of the software, but that could be that simply we need to work more on the AI side. So if we're going to do something like stratified screening with Mammatus as essentially as our gatekeeper as to whether or not they're assigned to a high risk extra screening or to just the regular screening, we have to look at what the cost is. And the cost is really how many more exams do we need to do to find another cancer? And this is sort of shown as a plot here, and along the bottom here is the number of extra cancers that we find, which we would have missed if we had just done regular screening. And then the number of screening exams that we'd have to do. So you could, in principle, just assign everybody to have additional screening, and we would find all those missed cancers in principle or in theory. But we'd have to screen everybody, and so the amount of extra screening would be 100%. We want to pick sort of the optimum value, which is hard to pinpoint, but we've sort of settled on a point right now where we're looking at roughly 25% of being assigned to high-risk screening, and that corresponds to potentially capturing or identifying 64% of those missed cancers, and that correlates to about 600 extra screens to find one additional cancer in a original screening population of 100,000 women. If you compare that to the dotted line up here, where you're looking at doing stratified screening based on breast density that is assigned by that qualitative measure from the radiologist, that A, B, C, D category system, that corresponds to about 1,100 screens to find that one extra cancer. So that suggests that using Mammatus would be a much more efficient way of doing this. We're not wasting all those extra screens to find those cancers, and we're capturing just as many missed cancers as we would have done if we had sort of screened everybody with dense breasts. So if this works, the idea is that we need to kind of prove this. So if we're to go on to a full-blown screening trial, you run into the thing that happens in screening trials is it's a numbers issue. And the problem with screening trials is that the disease itself is almost always rare. And I put this with exclamation marks and question marks because we always hear that breast cancer is the most common cancer in women and so on. And so it must be pretty common, and screening would catch these all the time. But the reality is that at any given time point, say when women go through for their screening exam, only 4 to 6 women per 1,000 women actually have the disease, and everybody else is completely normal. So getting the numbers of cancers to be able to show a difference between doing stratified screening and not stratified screening, you have to image a lot of women. The other parts to that too is this, this kind of analysis, the treatment that you get, even if we find these cancers earlier, may not be perfect. So some women are simply not going to respond to the treatments. And, is that because these cancers that we're now finding are just not treatable, or is it because the treatment's imperfect and maybe there's something that's not really showing a benefit, even though this could work if we had the right treatment? And so the question is this kind of stratified screening, will it improve outcomes? Will it improve morbidity and mortality? And you can do a power analysis, and you can look at the numbers that you would do if you were to do, say, a full-blown traditional randomized screening trial where you're looking at stratified screening in one group and then just regular screening in the other group, and the numbers come out huge. So in one arm, that's about 37,000 women to show essentially a fairly modest effect in the differences between stratified screening and non-stratified screening. So that's a big trial. Lots of sites have to be done. Lots of money would have to be set aside for this kind of thing. So if you're going to do that, you want to be as sure as possible that your stratification is effective and efficient, and that the other part too is that stratified screening is going to work. You don't want to go into this not knowing that stratified screening has a benefit, and you don't want to go into this with imperfect numbers or an imperfect masking risk evaluation tool. So in the pre-CATA program, where we're trying to establish that evidence, we're going to try to work with our partners and looking at trying to expand our information and our knowledge by leveraging existing stratified screening trials. And these are at Cambridge and Padua. And there we're going to learn whether or not Mammatus could more efficiently have stratified those women. Here, these trials are not using Mammatus. What they're using is that the breast density assigned by the radiologist, which we know is inefficient, and we can sort of re-subdivide those trial participants using Mammatus to see if we would have got better performance with that. The second group is to look at existing screening cohorts, and these are cohorts at Cambridge and Radboud. And these are just really large data sets where we have access to just an enormous number of mammograms, both with cancer and cancer-free, to know that Mammatus gives results that are informative and efficient and effective if we're then to use it as a part of a screening stratification program. And then finally, bringing it back home is to get some Ontario-specific screening data from London, Ottawa, and Sunnybrook. And that's really important because, each screening program is slightly different in terms of how the radiologists read, how many times they come or how often they come in for screening, what are the rules and criteria for detection and diagnosis and so on. And so we need to know that if we're going to do something here in Ontario, is it going to be relevant in our data sets? Because of the scope of this and the number of partners we have, it's really tricky to get all of these partners together and get information from them. And to mitigate that, we've adopted a federated validation approach for our Mammatus software. And the idea is to make it a little bit easier to get the REDs through and really to avoid really, long drawn out data sharing legal agreements to move data around. And so the idea here is that the software that is Mammatus is sent to each site. The imaging data that they have already stays at their site. It's under their control. we never even see the images, and it's all kept at the local institution. And then what we get is essentially reports of the masking risk scores or the radiomic feature scores versus the detection status. So, was the mammogram associated with a healthy patient that doesn't have cancer? Was it a cancer that was missed in mammography, or was it a cancer that was detected in mammography? And looking both at those screening cohorts as well as the stratified screening trials, and essentially trying to pull out that information that we need to show that Mammatus would work in a full-blown stratified screening program. So that's just showing here from each of the different sites where they're essentially housed at their own local locations where the server's used, and that the information flowing to us is essentially the summary scores from the masking risk program that is essentially collected by the clinical research groups at each site. So just a quick summary. The pre-CATA project is underway, and we originally had 5 sites identified. We've expanded that to 6 because Radboud University has a wonderful large cohort from the Dutch screening program, and so I think it'll be a wonderful opportunity to be able to assess masking risk in that data set. Our REB and data sharing is in place for 4 to 6 partner sites, and that's been a huge, long process to get that all in place, and we're going to have 6 of 6 very, very shortly, I believe, hopefully before the end of this year. And we are already deploying the code at 2 of the sites to essentially get it up and working in a so-called federated environment where we don't have a lot of access to it, and making sure that it works in a way that is going to give us real and informative data, as well as conforming to any site security and information risk analysis or risk factors. And so to summarize, Mammatus is a masking risk predictor for screening and mammography, and we've been using federated validation to try to see if we can demonstrate that there's enough evidence that Mammatus is an effective and efficient stratifier, and that stratified screening will have a benefit for women, especially for women with dense breasts. And that's the end.
Great. Thanks very much, James.